It is essential to note that each nucleated cell holds within its DNA the components necessary for their death through apoptosis. This is because programmed cell death is a result of protein kinase activity during which the proteins produced by one cell can initiate a response in other cells as well. Because it would be harmful for an organism cell to self-destruct through apoptosis without a requirement for it to do so, it is extremely important that these proteins are not produced in an unnecessary manner.
At the point when a mutation brings about a deviation from the routine of the cell cycle, and the mutated cell can multiply wildly, and the resulting tissue experiences metastasis, cancer can then occur. Cancer is the result of tissues that grow uncontrollably and spread throughout the body, which will eventually result in death. Cancer is known to be a threat because of the cell's ability to spread and the production of cells that results in the development of tumors. Tumors are a result of the amount of tissue that is grown in the body. During the procedure of cell differentiation, the tumor cells are not able to continuously divide as rapidly as expected because of the true association between cell death and cell division. The uneven course of action of cell division, uncoupling from the cell differentiation, and cell division brings about quick cell division in which the tumor develops quickly. There are two categories of tumors, which are benign, where a tumor just develops in a specific range of the body, and malignant, where the tumor can spread to different zones of the body.
P53 is activated by DNA damage. P53 activation leads to the inhibition of cyclin-dependent phosphorylation of Rb, which induces cell cycle arrest. BCL2 allows apoptosis to occur. P53 mutants have selective dominant negative effects on apoptosis, but not growth arrest in the humor cancer cell lines. A question has been posed as to what happens to apoptosis pathways and the growth arrest when both the wild-type 53 proteins and the mutants are expressed within the same cell. Since P53 ties to its DNA target arrangements as tetramers, and most of the P53 changes found in humor cancers are missense mutations, most mutants P53 monomers are fit for the oligomerizing with wild-type P53 monomers. So the possibility that heterotetramers development happens at consistent state conditions most likely expands because of the expanded half-life of mutants P53 contrasts with the wild-type 53 inside of a cell. The investigation of this dominant negative impacts in vivo has come to some disagreeing results. For almost all mutants contemplated, dominant negative impacts are seen just when appearance of the mutant's P53 proteins is in the surplus over the wild type. The study that I researched tried to determine if the differential dominant negative results on transcription regulation, growth arrest, and apoptosis within utilization of a novel bidirectional expression vector. The study observed that dominant negative effects are seen with just a subsection of the P53 missing mutants analyzed that this dominant negative effect has shown in the apoptotic procedure, however, has not on the grown arrest. siRNA. So, pancreatic cancer is a devastating cancer because of the limited amount of therapeutic options it has. The inhibition of expressions of target genes has been enabled by the use of small interfering siRNAs. Researchers have investigated how siRNA specific for BCL2 may be used as a therapeutic alternative for pancreatic cancer in vitro and vivo. As a result, researchers have determined that the BCL2 specific siRNA inhibits the expression of the target gene in vitro and in vivo. Anti-proliferative and pro-apoptotic impacts were seen in tumor cells, but were not seen in both the fibroblasts and the non-malignant tissues. So in conclusion of our presentation on cell death can be identified by two different processes, which are known as necrosis and apoptosis. And although these processes may seem similar, there are different keys that make up each type of cell death from each other. Apoptosis is known to be a programmed cell death and largely internal, whereas necrosis includes the loss of extracellular contents and plasma membrane rupture. Apoptosis is extremely important as it relates to the immune system because a lack of apoptosis can lead to cancer and other tumors, and the excess of apoptosis can lead to the loss of cells. And this concludes our presentation on cell death.